Hello Year 7, it's Ms Redmond here with your first summary PowerPoint for the cells topic, which is called How Can We See Cells? And it's all about using a microscope. If you haven't got one, please go and get a pen and a piece of paper now so that you can make notes and complete any of the little tasks that I give you as we go through. At the bottom of this slide, you can see the learning objectives for this lesson, so feel free to write those down. And you've also got a DNA here which says what is the smallest thing you've ever seen and how did you see it. So please pause the video now to complete both of those tasks. Organism is our first key word in this topic. An organism is a living thing and they are all made of cells. These are all examples of organisms. So we've got human beings, mushrooms, fungi, beautiful elephants. We've got a clownfish swimming around among the corals, which don't look too healthy. We've got some bacteria with their flagella. We'll come back to those in the unicellular organisms lesson. And we've got a beautiful lotus flower being pollinated by a bee there. So they're all organisms made up of cells. And cells are the smallest unit of an organism, and they're also known as the building blocks of life. Here are some examples of cells seen using a light microscope, which is what this lesson is all about. These are all examples of specialized cells, and we'll be looking at them again in the specialized cells lesson. These are leaf cells, these are red blood cells, this is a nerve cell, this is a human egg cell, these are human sperm cells, and these are root hairs. And we'll come back to those, as I said, in the specialized cells lesson. Okay, so how can we see cells? Well, we use a microscope. So here you've got a diagram of a microscope. And these are all the parts of the microscope. Please can you pause the video and see if you can match up the parts of the microscope to the correct labels on the diagram. So you can either point at the screen to remind yourself, or you could make a quick sketch of the microscope and label it up. The next slide is going to show you the answers. Alright, so here we have the correct labels. So going down this side, this is the eyepiece lens, that's what you look through. Then you've got the stage where you put your microscope slide, and that's going to be held in place using stage clips. Down here you've got the coarse adjustment and the fine adjustment, sometimes called the coarse adjustment knob and the fine adjustment knob, and the base of the microscope. On the other side, we don't often label up this part, but it's called the body tube. And then you've got the revolving nose piece, which is a very strange name. On the revolving nose piece are all of your objective lenses. Then the diaphragm can be adjusted to adjust the amount of light that gets in, and you've got a light source. On this microscope and on the ones in school, the light source is a lamp, but sometimes it's a mirror, so don't be thrown off by that. So how do you use a microscope? Now, this microscope is slightly different to the ones we use at school, because here it's the stage that moves up and down, whereas at school it's the body tube and all the lenses that move up and down. So the instructions are slightly different, but other than that, they are completely applicable. So follow the steps below to observe an object using a microscope. Move the stage to its lowest position. Now we would move the objective lens to its lowest position. Then, number 2, place the object you want to observe on the stage. Number 3, select the objective lens with the lowest magnification. This is a really key point because you have to get the object in the right place, which is quite tricky. And that's much easier to do with the lowest magnification, and it's much easier to focus on the object using the lowest magnification first. So that is a key point that they'll pick up on on exam papers. Number 4, look through the eyepiece and turn the coarse focus knob slowly until you see your object. That's another key point. You have to say, look through the eyepiece and turn the coarse focus knob, because if you don't, the examiner will think that you're just moving the stage up and down, or the lenses up and down, and not looking through the eyepiece, which isn't going to help you to focus on the object that you're trying to see. Then turn the fine focus knob until your object comes into focus, and repeat steps 1 to 6 using an objective lens with a higher magnification to see the object in greater detail. So what is magnification? Magnification is a measurement of how much bigger the object looks. Total magnification is equal to the eyepiece lens magnification times by, not added to, the objective lens magnification. Here's an example. What is the total magnification if the eyepiece lens has a magnification of times 10 and the objective lens is times 20? So remembering, we're timesing those together, so 10 times 20 is equal to 200. That means that your object is going to look 200 times bigger than it really is. Here's your summary. An organism is a living thing. A cell is the smallest unit of a living thing. We use microscopes to observe cells. Magnification is how many times larger an object appears, and the equation for working it out is total magnification equals the eyepiece lens magnification times by the objective lens magnification.